The workshop is designed in such a way that we introduce the very basic genetic concept for people who do a lot of imaging and have not been exposed to genetics before this. So genetics in actuality is very simple, and we designed it in such a way that if you understand general linear model, you will understand how genetic analysis works. Then we'll go into how to perform those genetic analysis with solar eclipse, and after that we'll have two advanced application talks. Lee reminded me that we have to be done by 5:15 because at 5:30 there's going to be a shuttle taking us all back to the Fairmont, so we may have to skip some of the Tom's slides, but let's carry on. So, modern genetics starts with the work of Gregor Mendel, who basically derived the laws of inheritance. He has been a very hard-working scientist who basically produced over 2,900 hybrid pea plants, and the work has been summarized in 1966 in a book called Experiments on Plant Hybridization. He proposed that what would basically be known now as Mendel's law of inheritance, or the law of segregation, or the first law, or the law of independent assortment of genes. So this is one of the illustrations from his book where he studied such traits as shape of the seed color, flower color, pod shape, and pod color, producing beautiful pea hybrids. In parallel, a second school of thought has been developing itself. Those are the so-called British biometricians. Mendel, if I remind you, was a Czech scientist who worked in Germany, and he was actually a monk. And British biometricians, the school of biometry was started by Francis Galton in also 1800. And chief scholars of that group included, of course, Carl Pearson, known as the correlation coefficient, and Walter Weldon. And this school was in competition to Mendelian school of inheritance. And two schools of thoughts were essentially addressing two different domains of genetics: simple and complex trait, inheritance of the simple and complex traits, respectively. This controversy between inheritance of simple or binary traits, such as pod color, or in human genetics it would be disorder type, and inheritance of complex traits such as color of the skin, or color of the iris, or heritability of the height and weight, was resolved amicably without any physical alterations between two schools in a seminal work by Ronald Fisher, who is a father of quantitative genetics. And this seminal work was published in the Transaction of the Royal Society of Edinburgh in 1918. This work is known as the Fisher's Bridge. This is a reconciliation between the Mendelian segregation of genes and traits with the determination process of complex traits. Fisher's model assumed that complex traits were determined by a large number of genes, each of which exhibiting Mendelian segregation, and each of those genes providing a small additive effect. And this is the first time the term variance was coined here in terms of the genetic analysis. So this is basically the foundation of the modern day linear model, along with its use of the normal distribution in genetic analysis. So all of the modern genetic analysis can be traced back to this paper. So this is basically an example where we're looking at the skin pigmentation and inheritance of certain polymorphisms. and that our trait distribution, such as darkness or pigmentation of the skin, follows this normal curve with different loading on a discrete set of genes. So we are going to, throughout this presentation and throughout this workshop, we will be only talking about normally distributed traits that have some variance. If your trait is not normally distributed, Solar offers powerful transformation tools that will ensure the normal distribution for your trait, and this point will be addressed in the second, the follow-up talk. So, what the genetic analysis does, in a very simple terms, is variance decomposition, not unlike the imaging analysis that we all do in terms of functional analysis of functional data, or DTI data, or structural data. Basically, we decompose our variance into two distinct components. One of them is due to genetic sources, and one of them is due to environmental sources. Those two together add to the total phenotypic variance that you observe in the trait. Moreover, genetic component of the variance can be split further into the additive genetic variance and the dominant genetic variance. The additive genetic variance indicates that if you have two genes of the same kind, the phenotype value will be doubled. Dominant genetic variance, so basically it's an additive recessive disorders versus dominant disorders. In the dominant variance, a single variant of the gene ensures the 100% of the phenotype. And we are particularly interested in this additive genetic variance because that's the one that is inherited in most of the polygenic disorders. So here, for example, we see a very nice example of additive genetic variance. You have a single allele with variance AA, AB, and BB. So when you have two AA alleles, you have a phenotypic trait of -A. When you have intermediate allele, you basically have a trait of, you have a phenotypic value of D, and a BB allele lives with a value of A, in positive A. In this case, of course, D is equal to 0. So that would be a perfect additive effect. So in this case, the heterozygotes is halfway between the two homozygotes. And the important part is there is dose response effect. So additive genetic effect can be further parcellated into the probability of those individual alleles. So you have a probability P and Q for those alleles, and then you have the effect size A, D, and then you can also calculate the dominant genetic effect. It's a very simple, probability type equation. In a more advanced analysis, you can actually have an epistatic term, and while it is important to model for epistatic interaction, we don't typically do it in most of the analysis. So, in case of the epistatic variation, you have an interaction variance that exists when A or D is a function of the genotype at another locus. That's what epistasis terms that I think was very popular about 5 or 10 years ago. Everyone was talking about epistasis, which basically means that when you have a variance in one gene, it actually affects variance in a quantitative trait expression for another gene. There is further parcellation can be achieved by parcellating environmental variance into the common environmental variance and unique environmental variance. So the common or shared environmental variance is the C that is commonly seen in the ACE model, where A stands for additive genetic variance, C stands for common or shared environmental variance, and E stands for unique environmental variance. So common or shared environmental variance is explained by the extra variable you put into your model that accounts for the family environment, for a family or household environment. Now, this can be a very powerful approach when executed correctly. However, in most of the instances, that is not executed correctly. I think to fully execute ACE model, you actually have to have genetically close individuals who live in different households, such as children who have been adopted, or such as incomplete families, where you have a split between families, such as, for example, father or mother leaving a family and moving to another household. Only then when you can start accounting for the common or shared environments. Just indiscriminate use of the ACE model may actually lead to C components borrowing some variance from the A component, because you're basically having two genetically similar people living in the same household, and so you have this interaction between genetic similarity, which we shall call kinship, and environmental similarity, which we call household effect. Now let's define one of the fundamental measurements in genetics, and that is heritability. I'm sure you have heard heritability term used many times during the talk. So heritability is the proportion of the phenotypic variance in a trait that is attributable to genetic effects, and that's broad sense heritability. In this definition, we actually use narrow sense heritability, where we're only using the additive effects of the gene. So it's only the additive genetic effect is what we account for heritability. So it's kind of strange. Anytime you actually see heritability in the paper, that's a narrow sense heritability. Anytime somebody talks about broad sense heritability, they have to say it's a broad sense heritability rather than just heritability. So here's the broad sense heritability. In the broad sense heritability, we are only interested in the total genetic variance as a proportion of the total phenotypic variance. So it's a very simple model where you just calculate this variance, you calculate total variance, so obviously you know the total variance, you take one over another, and your h square is going to be somewhere between 0 and 1. 0 would indicate that total genetic variance is 0, and so there is no genetic variance. 1 obviously would indicate that the entire phenotypic variance is due to genetic sources. So it's very common to get heritability value of 0 or as near to 0 as making no difference. If you typically get h square value of 1, it typically indicates that you have some problem with your model or trait, or something's just not working right. But you can get h square values pretty high, especially with imaging measurements such as head size or brain volume. H square values are typically 0.9, between 0.8 and 0.9. So this is the narrow sense heritability, where you only look into the additive genetic component over the total phenotypic variance. How can we model the phenotype? So this is actually a very simple general linear model, where you have a trait, which we call the phi, P as in phenotype, and then you have a baseline mean, then you have coefficients for fixed regression factors such as age and sex and disorder type. Then you have term that is responsible for additive genetic effects, and then you have a term that is responsible for random genetic effects. It doesn't look any different than any GLM model you design for your functional experiment, where you put covariates as well as you put AY value, but in A value it's actually going to be your stimulus. So it's very similar in that regard. And then, just like you do it with GLM in imaging study, you model the phenotypic covariance. And you model it by the means of two important matrices. A matrix of phi and a matrix of I. Matrix of phi corresponds to the additive genetic variance, and matrix of I, which is actually a unit matrix, or matrix of just 0 and 1s, models the variance due to unique environmental influences. The matrix of phi is filled with the values that correspond to how much DNA people in your study share among each other. So it's a matrix that's symmetric across the diagonal. Diagonal is symmetric matrix, and all the diagonals are filled with 1s. If you have monozygotic twin pairs, they also share 100% of the variance, so you put 1. Parent offsprings and siblings share 50% of the variance, so you basically put 0.5. Grandparents and grandchildren share 1.25 of the variance, first cousins and second cousins, and so on and so forth, until you actually get down to people, just randomly chosen people, like what we have here, who will share actually about 1 to maybe as much as 5% of the variance, especially people from the similar geographic locality. So there is a big move right now towards so-called empirical kinship matrices, where we take subjects who are assumed to be completely independent, and then we estimate the degree of relatedness among them by looking at the variance in their genotypes. And amazingly enough, variance that we have in a trait can be contained in this circle, so to speak. And this trait is controlled by 4 distinct genetic factors. And each of those genetic factors explain that much of the variance that corresponds to the area under each circle. So in this case, the heritability will basically be the area under each of those genetic circle factors divided by the total area of this circle. Perfectly sensible. Now, what if some of that variance can be absorbed by covariance such as age and sex? For example, let's say this is height. The trait is height, and this is our total variance in the group. We're interested in how 4 genetic factors influence height. But we also know that age and sex will greatly attenuate the height, right? We are born fairly short, and based on our gender, we have, gender-specific dimorphism. So males are typically taller than females. And if we just account the total variance, we will be doing ourselves a disservice, because in actuality, fixed factors such as sex and age can explain a fair bit of the total variance as it is. So by using age and sex as covariance, we can actually estimate a truer degree of genetic dependence by subtracting the variance from age and subtracting the variance from sex from the total model, thereby producing a more coherent and more accurate estimation of heritability. So this is done as a part of the sporadic modeling. You basically perform the null hypothesis testing, where you set all the regression coefficients to covariates to 0. And then you repeat this model when you allow the covariate coefficients not to be 0. And you do it for every single covariate. And so you can estimate the total variance explained by each of the covariates, decide whether that variance is significant or not. And if it's significant, then you include that covariate into your total model, because including all the covariates in your total model willy-nilly may likely undermine the model by introducing extra degrees of freedom. So it's just a standard GLM analysis. It makes all perfect sense, except you have to use it with caution. In general, you only want to use covariates that are fixed factors, so demographic or environmental. So, for example, if this trait is height, and we know that BMI is also a measure of height, in addition to the measure of the weight, it's a ratio of height over weight, then this BMI may actually influence or explain the variance by genetic factors 1, 2, and 5. And by using this BMI as a covariate, you're actually going to reduce the heritability value by the corresponding fraction. So if we include BMI as a covariate, we reduce the power to detect the effects from G1, G2, and G5, and genes that influence both BMI and the height. And so it always pays to run your model with and without covariance. So specifically covariates like a disorder, those can be very much power robbing from your heritability, especially if you're looking at a disorder-specific trait. So if you observe that adding an extra covariate greatly reduces your heritability, this is a good time to reflect on what this covariate is and perhaps use a more advanced genetic analysis to estimate this effect. Now, we have looked into the heritability analysis, right? So, you heard the trend GWAS, or genome-wide association analysis, many times during the last couple of days. So, what is that GWAS analysis? Well, in actuality, it's very, very simple. In the GWAS association analysis, you use a genotype or allelic frequency for that particular individual as a fixed covariate effect. So you basically measure genotype association and whether the trait mean differs by genotype, taking into account the non-independence between family members. So, for example, what if G4 is actually a polymorphism, common polymorphisms known as A39T? So for the GWAS analysis, you're only interested in the variance that is explained by that particular polymorphisms in relationship to all of the variants minus the variance explained by fixed covariates such as age and sex. So GWAS analysis, or what it does, it measures QTN, or quantitative trait specific heritability. So H squared QTN, and that's what the power of the association, that's how they measure the power association, that's what powers those Manhattan plots, is the variance explained by this QTN by the total variance. So additive genetic variance can be modeled as QTN based on this simple equation that we looked before, where you have a particular genotype, three particular genotypes and options, and you can basically estimate the heterozygosity and the effect size of that particular QTN. So typically in those analyses, the effect of the QTL is modeled as additive, and you basically say that genotype AA corresponds to covariance of 0, genotype A, capital A, small a, value of 1, and AA is the value of 2, and that becomes your covariate vector, which is also a genotype vector. And then you perform the association analysis, and the power for the association studies, of course, is a function of the sample size, the family configuration, and the QTN variance, and the linkage disequilibrium between QTN and a genotyped marker. So when we genotype with GWAS panel, we typically genotype with a very coarse resolution. Typically, each marker is separated from the other by millions of bit pairs. That means that our hope is that that particular SNP that we study actually is reflective of the true genetic material nearby, and so it's in linkage disequilibrium with the SNP of interest, or the SNP that we actually have on hand. So this is, for example, example analysis of 46 CT genotype for the protein level FX2 levels. So we have for the CC genotype allelic polymorphism, we have FX2 levels of 128. For the CT, we have level of 92, and for the TT, we have a level of 55. So we have a perfectly additive effect of this polymorphism that is highly significant. So in fact, if we plot the FX2, which is also known as the prothrombin level activity by the subjects with a particular genotype, we will see this nice step-ladder distribution with about equal difference between the phenotypes, suggesting that that's a perfectly additive effect. Linkage analysis. So far we looked into the genetic association analysis. In this case, we are basically saying that all subjects who have the same genotype should have similar phenotype, and therefore subjects with different genotypes should have different phenotype. Very simple type of analysis. This is called identity by association. Linkage analysis do a much more clever hypothesis. What linkage analysis say, it's not whether the subjects have the same genotype, it's whether the subjects receive that genotype from the same ancestral source. There is a big difference between them, right? So in one case you basically say, well, I have so many subjects, I want to know whether their genotypes are the same, which could be due to random ascertainment of the subject, or could be due to many other factors. A much more coherent analysis would be saying that I want to look into the subjects who received the genotype from the same ancestral source. So they carry the same piece of DNA that they received from the same ancestral source. And that's what we're going to represent by a matrix pi. And sigma and variance due to this matrix is going to be sigma QTL. So this type of analysis is known as identity by descent. To restate it, this type of analysis hypothesizes that the proportion of alleles are shared by a relative pair that derive from the same ancestral source, or identity by descent. This is, of course, in contrast to allele shared by sort of identical by state, which is the GWAS analysis, in which the ancestral source of the allele is not considered. So IBS, or identity by state, is association, and identity by descent is short for linkage analysis. So to facilitate those analyses, you basically have to make this matrix of pi coefficients, where pi ij would be the IBD probability for individuals I and j to share the same allelic, or the same genotypes from the same ancestral location. And we can apply the same variance component-based linkage analysis to the same situation, and in a region containing a QTL that influencing the trait, relatives who are phenotypically similar will share most alleles that are identity by descent than relatives who are phenotypically dissimilar. Now, this is a sort of a vintage definition of the identity by descent. Right now, we can actually facilitate this type of analysis even in unrelated individuals. Because after all, we as a species actually have a very small genetic variance among us, because in at least in 2 points of our evolution, we were limited to a very small number of subjects. So we all have the same evolutionary image and the same so-called linkage analysis. So we are basically looking at how a particular trait is going from one generation to another, in comparison to the family structure. So here, for example, father had a particular trait, and that particular trait was transmitted to three offspring. Here we have two offspring with a trait and no trait among the parents. And here it's a much more complex pedigree structure. And again, we have sort of a random showing up of the trait in the offsprings, but they're not present in the previous generation. And overall, using this pi matrix, we can describe this relationship fairly completely and then plug it into our general linear model and see what variance is being explained by this structure. So here you can actually see that by having this super pedigree, we can actually observe that this trait is probably propagating from way before ancestral, upon the ancestral stream. Okay, so now let's actually see how we can perform those analyses in software, unless you have any questions. Yes?
I had a question. I was contemplating doing a pedigree study of a large database of about 50,000 subjects, clinical subjects. We have an observation of certain traits being shared by first-degree relatives. For instance, ADHD parents can have certain traits that they share with their children. And we can demonstrate this by the brain scan. I think what I infer what you're saying is that if the only information I have is the family relatedness, then I would be impeded in discerning the effects of that relatedness from the environment for first-degree relatives.
I'd say not. I'd say not. So it's you can answer that question directly by assembling your pedigree and running heritability analysis, and then introducing the shared household effect and see where that will take you. Likely what's going to happen is, if it's not a true household effect, you will get some hit on the heritability, but the household effect will be not significant because it will have a huge confidence interval. In which case you can basically say that it's very unlikely that the shared household effect is playing a role in here. Simply because it will have a huge household, it will have a huge confidence interval, because most of this effect will still be attributed to the additive genetic effect. And additive genetic effect will be much more closer to the transmission of the trait, because you will have some siblings who have that feature, right?
Right.
And some siblings who don't have that feature.
Oh, okay, yes. So
and they all be living in the same household.
Yes.
So it's very likely that what you will get is that your heritability is very high, and the household effect will be, while they take some piece of heritability, you will have a huge confidence interval, because it's not as close to the effect itself, because you can have two siblings with different traits living in the same household. And that especially works really well for quantitative traits. Traits that are present, to some degree, that can be present to some degree in the two siblings. So especially if you have more than, if you have families with more than two siblings in your design, then it will be very easy to parse out the additive genetic effects from the common shared environmental effects. Especially if you have sort of variance among the families where you have one sibling with the trait, two siblings without the trait, or two siblings with the trait, one sibling without the trait, or all three siblings with the traits. Because they all live in the same household, they all share the same household, but that household cannot explain this parcellation of the trait among subjects. So if you just put it all into the general linear model, very likely your variance parcellation will be heavily tilted towards additive genetic effects than if they are true additive genetic effects. And if you have 40,000 people, I'd say this is going to be a remarkably powerful study. I mean, Solar has no problem working with kinship matrices as big as 500,000. So you can invert those matrices fairly easily. So you can basically do those analyses in Solar fairly easily.
Okay. And will my model be helped by having second and third degree relations?
It would be very helpful to have those, but commonly you don't ascertain them, right?
What's that?
But do you ascertain them? The third and second degree relatives, do you actually have them ascertained?
Yes, in some cases I can, yes.
Oh yeah, then of course,
Yeah.
the more the merrier.
Yeah.
So the more variance you have in the kinship matrix, especially at the range where you have values between 0 and 0.25, then the stronger your model will be.
Oh, excellent.
Yes.
All right.
Are there any more questions? Yes.
So, from what I hear, Solar is basically operating on, it's not deriving a genetic variance from the data itself, so it's assuming it's purely based on the kinship matrix, correct?
To some degree. So there is a way you can, so you basically have to decide, it's more like experimental design. How do you want to ascertain the relatedness among your individual, among the members of your pedigree? So if you wish to code the pedigree directly, and that's what typically done, then I'll be explaining to you how to do it in the next hour. It's actually very simple. If you wish to ascertain pedigree by other means, such as empirical relationship between them, then Solar allows you to simply use a kinship matrix of your own design. And then you can basically do whatever you wish. As far as getting the different kinship matrix, this is something that we are still considering to put in, because the methods, there is such a variety of methods out there that we are waiting for dust to settle, so to speak. And in many cases, I think this will be done as a shared project. So if you or anyone in the audience have an interesting proposition for a study and is willing to put some effort into it, then we'll be more than happy to help you with sort of performing that study in Solar, because it's of great interest to us as well, and hopefully the methodology will be useful to others. So, for example, the last part of this workshop, I'll be talking about very fast heritability estimators, and that was exactly developed as a part of the collaboration with Tom Nichols, who needed very fast heritability estimates for his permutation analysis, and we worked together and we are putting them all into the Solar for everyone else to use. Alright, shall we go to the second talk? The this is my talk. So, in this talk we'll go through a historical perspective, then I'll be talking about downloading and installing Solar Eclipse, then creating a Solar Eclipse directory, including pedigree files, marker files, and phenotype files. Then I'll go through the most common analysis types, specifically univariate and bivariate analysis of variance, linkage, and association. And I'll be talking about some new methods that we implemented since last year and how we can use Solar Eclipse for mega and meta genetic analysis and perform additive genetic analysis of credibility, as well as analysis of fixed genetic factors association in the studies that are composed from diverse data sets. So for example, you have your own data set and then somebody else gave you another data set and you want to combine those data sets together. How can we do that? And that's exactly what I'll be talking about. So Solar Eclipse is the software tool for imaging genetic research. We developed it for multi-platform and we compiled it for multi-platform, which includes PC, Mac, and Linux. But as somebody already asked me, all our development is done under Linux, and we compile PC and Mac version typically under request. We have a lot of requests for the Mac version. We've never had a request for a PC version, maybe once or twice. But if you have an interest in a PC version, do let me know. But otherwise, we primarily work on the Linux, and we can also accommodate other Mac. So it supports a wide range of genetic analysis of both discrete and continuous trait. It supports polygenic, quantitative trait linkage, GWAS analysis in related and unrelated individuals. And it supports univariate and multivariate genetic analysis, which is a bit of a rarity. And supports discrete and continuous covariance. And it implements functionality of the free ancient genetic tools that have been used by the genetic field for decades, and that's Mendel, Fisher, and Search. So Mendel and Fisher are the classical genetic analysis that were developed in 1960s, and they were developed in UCLA, University of Michigan, and Ann Arbor. And I think since then they have pretty much gone into ether, so to speak, but we made sure that Solar has the full functionality of those two packages. So Mendel is sort of a historical relic that was originally intended for gene mapping calculation and QTL analysis. And this was the analysis that basically was used to discover such genetic disorders as cystic fibrosis, multiple dystrophies, and many others. So it's a very sort of, well-nominated analysis tool that has a good legacy that goes over several decades of successful research. Fisher is the genetic analysis of quantitative trait that specifically to perform variance component modeling of the polygenic inheritance and environmental factors. And it provided the functionality for calculation of heritability and genetic correlation. So Solar basically took the functionality of Mendel and Fisher and combined them for the use of genetic likelihood correlation. It assumes that the variability in trait is explained by multivariate normal distribution, and therefore it supports both multivariate and univariate genetic analysis. Also does proband correction and robust outlier detection. And maximum likelihood estimation, that's the engine of the variance component, is a routine called Search, which is an old algorithm that is very robust, albeit quite slow. So we use that search algorithm as the maximum likelihood engine for the Mendel and Fisher. And it was optimized to work with user set bounds and limits, and sort of it maximizes the cost function and samples a function over the grid, and implements weighted least square and user-defined estimators. And John has optimized it for discrete traits and rechristened that algorithm Optima. So what is solar eclipse in sort of, in the diagram? So we have nifty libraries, standard nifty libraries that we can use as an input. All the higher level functionality is run in an R-like shell and supported by TCL interpreters. So TCL interpreter is actually built into the Solar shell, and you can set all your TCL, you can use your TCL commands. I think eventually we may want to rewrite this part in Python, because Python has become much more fashionable than TCL in the recent days. So this part may have to be redone. Then there are C++ libraries, and that basically handle all the low-level processing. And the advantage of TCL back then was that TCL has a very nice integration between C and C++. So you can write all your higher order genetic algorithms in TCL while calling on the functionality in C++ directly from TCL, very much like Pythonish way it is done now. So progress since last year has been fairly brisk, I have to say. So we now have a polyclass function that's now standard, and that allows you for a combined mega or metagenetic analysis of multiple pedigrees. And Netatalk will show you how this can be used for GWAS analysis. There are also some changes in data handling. The inverse trait normalization can now be performed before or after covariate regression. That was a request we got from somebody who participated in the last Solar workshop and asked specifically for that functionality, and we made it available. But we also now developed Solar Eclipse web version, where if you have a project where you have related individuals and you would like to have an analysis portal where your users can come in and say, well, I'd like to estimate heritability for this trait, or I would like to do this genetic analysis, they can actually do it directly from your website. So we have a web version of Solar to power sort of analysis portals for individual projects. And we are now working on the great speed up, specifically to accommodate the change, the sort of full scale change in computational intensity for the whole genome sequence analysis. Those of you who worked with GWAS may have been overwhelmed with analysis of 1,000,000 genotypes, albeit typically it's just 700 genotypes, because a bunch of genotypes typically don't pass the QC well. Think about it in terms of analysis of 3 petabytes or something like that. You basically need a completely new level of computational efficiency to accommodate those analyses. And this is driven in part by availability of the whole genome sequencing, and in part by the fact that most of the projects nowadays actually impute their GWAS data to the 1,000 genome standard. So instead of having just one genotype, they have 20 to 30 million, 1 million genotypes, they have 20 to 30 million genotypes. So you can imagine if your GWAS takes an hour to perform, then performing a GWAS analysis in imputed data will be quite a bit longer. So that's why we are putting extra effort to develop single-step estimators of heritability and association, and that's the last part of this workshop, as well as doing GPU acceleration. We're also now working on video tutorials for solar eclipse. We have a very nice lady who basically learning solar, and she describes her experiences of learning solar in the videocast tutorials, which you're welcome to peruse on our website, where she talks about stuff like creation of the pedigrees, or performing genetic analysis, or looking through the data directory and making sense of your results. So downloading and installation is fairly simple. And the only thing we ask is for you to get the software key license, and you just email to this email address and you'll get it pretty much right away. We have website. Also, technically solar is open source project. So therefore you can request source codes from us. However, we are very hesitant to distribute source codes because in the past we have some individuals who requested the source code and then they spent several months accommodating their development, and then nothing came out of it. And they sort of said, nah, we give up on on this, we move to better things. And it's like, well, we spent 2 months with you on hand holding you and showing you how the makefile works. So if you want source code, please at least have experience in compiling large multi-platform enabled C++ code, and don't ask us where to get this library or that library for your particular line of distribution. We simply can't put time and effort into this. So that's why we prefer to very much work on the project basis, where you ask us to do some changes to the solar code, and we'll accommodate, because technically it actually gets to be a lot quicker and more effective than just giving the source code away and letting you go at it. But if you just want the source code away and go with it, then by all means do it. Just we can't support extramural developments quite yet. So when you install the solar and you type solar, the TCL interpreter starts. And when you type a command like polygenic, which is a very sort of ubiquitous command that does the polygenic modeling, the TCL interpreter reads a solar.tcl file, which codes for all the popular functions such as polygenic. And then it executes the TCL function called polygenic in the solar.tcl code. And the solar.tcl code is actually freely readable with any text editor. So if you're interested in modifying the upper level genetic analysis, by all means go at it, because it can be done very, very easily, and I'll show you how to do it. So this polygenic function, it basically analyzes the arguments and existing global variables. Then it chooses among several analyses, such as univariate or multivariate, and calls upon compiled work for computationally intensive procedures. Very straightforward. So for those of you who don't know what TCL is an interpreted language that was quite popular until maybe about 5 or 7 years ago. It stands for the tool command language. It's very similar to shell scripts such as bash and sage, albeit it's actually a properly vetted programming language. And all solar commands are TCL functions. So if you have a desire to modify a particular function, what you can do is you can type show proc and then command. And this command can be just, polygenic or any other command. Or you can just type show proc and then the name of the command, and then call new command.tcl. And what it will do, it will take that particular function out of the solar TCL code and save it into a new file called new command.tcl. So it will create a copy of the original TCL script in your directory, and now you can open it with the text editor to see what it does, and you can rename the function, and the next time you run solar, it function out of your home directory or out of your solar directory, from the directory where the solar was executed. So solar works in directories. And so all projects are organized by directories. I know it's a very Linuxy type environment, but that's what we sort of grew up with. So Solar reads the default files from the directory it started from, and that basically includes pedigree and default phenotype files. That is not to say that you can't reset, parameters in the Solar to work at different directories, but I even forgot how to do it, so typically I never bother with that. So the directory should contain the pedigree file, also known as the kinship matrix, and that's the file that defines the family ties among the subjects. And we definitely have a video explaining how to code the pedigree file on our website. And if you have any questions, by all means contact me. I probably coded in over 100 of pedigrees in Solar, so I'm quite up to date on all that. So coding the pedigree in Solar is actually very simple. You simply make an Excel file and save it as CSV file. And 99% of the questions I get asked from the users is typically due to one simple problem. When Mac users save the pedigree file from Excel, they should save it in the Windows CSV format, because Mac Excel for some reason uses some really weird end-of-line characters which are not interpreted correctly by the Linux. And it actually, right now we had so many requests for this that we actually now have a function in Solar that will analyze your file and then record it into the Windows format. But it's easier to save yourself trouble by simply choosing the Mac CSV. In Mac, when you save it, just choose Windows CSV format. And pedigree file must define subject ID. And subject ID is later going to be used for your phenotype files, and that's the way you link your pedigree to your phenotypes. And then you include IDs for fathers and mothers, and that also includes founders. So for twin studies, I know many other packages like OpenMX do not actually make you code the father and mother. In Solar Eclipse, we actually ask you to code for mother and father. So you have to code your pedigree all the way up to founders. And founders are the subjects who don't have mother and father and who are omitted from the analysis. We also need to code for the gender. You code 201 or M and female. Everything else is pretty much optional. You can code for the family ID or the household ID. So if you want to do common environmental influences, that's how you would do it. You can have a monozygotic twin label. And that's important because those are the only people in the pedigree who share 100% genetic variance between each other, among each other. So monozygotic twins label should be there. And another optional parameter is called class, and that's used for megagenetic and metagenetic analysis to basically separate, to give Solar some knowledge that the data actually comes from separate pedigrees. So again, each active subject has to have an ID, family ID, gender, and parents' IDs. And those additional variables include class and household, or family ID, which specifies the common environmental influences. Oh, where's my? There should have been a video there. Oh well. Now we'll have time to watch it anyway. So here's the example of the pedigree for UCLA twins. So we have. That's a QTeam study that NEDA has published. NEDA's group has published probably several dozen papers already. So you basically define an ID. That ID can be randomly selected ID. And you see, those are the IDs for active subjects, because they have IDs for father, FA, and mother, MO. And then they have gender, and then they have family ID. And because this is a twin study, we have several, or many, monozygotic twins. And this is where you code the founders. This is where you define the parents for a twin study, for which you actually don't do the analysis. And Solar actually can get very grouchy, because we have many extensive checks on the consistency between genders for the twins and genders for the mother and father. And so it's very common for people to start getting messages such as, Solar will be complaining, Solar will be rejecting your pedigree because you have monozygotic twins of different gender. Well, that clearly cannot happen. And also it will reject your pedigree if all of your parents are of the same gender. Unfortunately, Solar is not politically correct this way. We do not allow for the same gendered parents quite yet, because in terms of genetic analysis, that doesn't make a whole lot of sense until they actually find a way where they can clone either sperm or eggs. And at that point, I promise we will allow for the same gendered couples, because it will become meaningful. And so here's how you run it. You basically run Solar, and it's actually an old slide. So it sort of starts like this, and then there is a prompt. Kind of, if you used R, this is going to be very familiar to you. And the Solar shell actually inherits all the shell commands below it. So if you type ls, it will simply bypass this command to the shell that's below it, and it will show you all the files in the directory. So here you have gobs underscore, you type ls, you have gobs underscore ucla underscore pedigree.csv. And it has a sort of name pedigree, so you may say, well, that looks like my pedigree file, so why don't I load my pedigree file? And the command to load your pedigree file is load pedi, you see they're separated by space, and gobs underscore ucla dot pedigree.csv. And you see, in terms of the TCL input, load is the main function. And what goes behind it are the parameters for the function. So load is the function. And again, it's coded in the same simple format where you have two IDs and shared variance between them. So the new functionality in Solr allows you to take the phi2 matrix that is generated by, for example, empirical kinship procedures and create a pedigree based on that kinship matrix. And that functionality is implemented by the name of matrix, where you can actually load a matrix and then assign that matrix as a kinship matrix, and then a new pedigree will be generated. Also, we support import from many other genetic analysis packages, but I don't think it's very important here, given that most of them are very specific and nearly obsolete. So creating a SNP file is actually very simple again. Solr uses a simple marker format where we have CSV or csv.gz file with a header. And again, you have ID or ID column that defines the subject IDs. And then you basically, if you put your SNP names as RS, the Solr will know that those are actually not strictly speaking covariates, but rather SNPs. But it does interpret them as covariates as well. So if you want to do just a genetic analysis on a single SNP, you basically take that SNP and include it as a covariate. And when it runs the polygenic model, it will tell you the proportion of the variance explained by that covariate, such as SNP, and assign a significance to it. But if you type NGA analysis, it will actually look for the all covariates that have RS name in them and perform the sort of GWAS analysis. We're still working on the Plink genotype files, and again, it's work in progress. Trouble with Plink genotype files is that they're designed to be read in full. And that now creates a big problem, especially with the imputed data. So Plink, for instance, can't handle imputed data because the file format requires reading of all genotypes in full before it becomes meaningful. And since we are now, half of our analysis are done in either imputed or whole genome sequencing, we are now thinking about a completely different imaging genetics-oriented genotype file format, but that's different now. And you can now, let's go to creating other phenotype files. Phenotype files, it's same as the covariate files, same as the marker files, are the same CSV files, except, the format is exactly the same. Solr phenotype files are basically CSV, comma-separated format. And it has to have either ID or EGO to designate the identifier for the ID field. And typically, file for a continuous trait goes like this. So the first line is the header, and that defines the trait name. And so it's ID, age, sex, chromatopigment, phase flare, and then you build up the table like this. In addition to just float numbers, phenotypes can incorporate imaging data. In this case, they need to be specified as to us, and I don't think anyone else really does it so well, is the meta and megagenetic analysis functionality. Because Solr engine is so flexible and powerful, we can work with pedigrees of random length and complexity. So we can work with family data, twin data, unrelated data, and separate pedigrees can be combined at the raw data state. So you can merge several small populations into a synthetic pedigree and perform genetic analysis as if working in a large extended family. So the advantages of megagenetic analysis is that you actually have much more powerful analysis degrees of freedom-wise than meta-analysis. And the test of significance is therefore performed on a high degree of freedoms. And smaller sample, and therefore smaller samples can be combined into a single data set with pretty much additive increase in the power. And this makes the results less weighted by the discrepancies in subject numbers and less affected by the variance in standard deviations among the cohorts. And this can be used for also testing of the heterogeneity of genetic effects. But it also has, it also outputs, as a part of this analysis, it also outputs the measurements that you can use for metagenetic analysis. And the advantages of metagenetic analysis are that they're easier to perform and they're more readily accepted for publication because meta-analysis is basically the gold standard of the multi-site studies. So how does one proceed with those type of analysis? It's very simple. You can catenate pedigrees just using a different class field. And class field defines all subjects belonging to the same pedigree at the raw state level. Sporadic model, or the model that fits the covariates, is actually executed separately for each class because it's common for us to assume that the covariates are going to be site-specific. So therefore, you, for example, can combine samples of vastly different age range. So you can analyze together data from young subjects versus old subjects, because ideally the effects of age discrepancy and between cohorts will be addressed by basically covering those effects out per cohort. Same with the gender, same with the disorder. So after that, the individual data sets or residual of the individual data sets inverse Gaussian normalize. And so we are combining data by ensuring equal distribution and variance in the trait. And in fact, we are enforcing equal distribution and variance of the trait. And then we perform several polygenic traits, specifically a test of significance for each pedigree, crosswise test of difference in genetic effects, and test of significance in a combined pedigree. So basically you get outputs for meta and megagenetic analysis. For this part, we developed the polyclass function. And again, you can see what is inside this function by simply running that show proc command. And the polyclass function will model additive and fixed genetic effects across pedigrees. And it is performed per pedigree, so it performs per pedigree modeling of genetic effects and performs combined modeling of the genetic effects. So, example of mega association analysis will be presented by Neda, so I'm just going to skip through that altogether in interest of saving time. And if some of you guys have your own large projects where you want to sort of showcase your results and allow your users to perform genetic analysis directly from your website, just contact me and I'll try to accommodate you with the Solar Eclipse version. So large projects can put Solar Eclipse front end to their website that provides for sample analysis. And users can basically execute the full functionality of solar analysis without having access to raw data. So it's sort of done in a secure environment where all the internal variables such as pedigree and structure is stored on your website and not distributed, and providing immediate access for the discovery or replication sample for your users. This was done, for example, in collaboration with Human Connectome projects who are using now Solar Eclipse as their front-end genetic analysis tool. So you can choose So if you're a registered user of Human Connectome dashboard, and I have to warn you right now, the dashboard is actually in the state of disarray because they're moving from 200 sample to 500 sample, and that creates a whole lot of uncertainty. So you basically choose your trait, then you designate your covariates such as age and sex. You choose an option for inverse normalization, and you hit a program button, click run, and this gives you a full output of genetic analysis of heritability. In this case, it's for the FA for the body of corpus callosum, and you can see that in the Human Connectome sample, the heritability of FA values for the body of corpus callosum is 0.84, so it's very high. The standard error is very small, and therefore the significance is just huge. And the effects of the covariates, such as age and sex, are really not all that significant compared to the significance of the heritability, especially age, which is basically robustly non-significant, of course by design, because Human Connectome project only recruited subjects aged 25 to 35 as to avoid any age effect in their phenotypes. Very clear about that. So right now our emphasis on development is a great speed up, so to speak, in preparation for whole genome analysis. So the idea is to speed up the processing by 1,000,000-fold. I know it's a tall order, but this is necessary to allow for real-time GWAS analysis for solar eclipse web version in projects like Human Connectome projects, and allow for the whole genome sequence analysis. And it will make use of the GPU technology, because this can greatly reduce parallel processing costs, since those GPU cards allow you to purchase processing capabilities at less than $1 per processing core. And one part of this great speed up is going to be in the last talk of this talk, and that's single-step approximation. And so I won't go into this right now, since this will be described later on. And another part of this is the GPU processing. So that is and this is done in collaboration with doctors DeMotta and Frolin from NVIDIA. And GPUs allow for massively parallel computation capabilities, such as voxel-wise heritabilities, or GWAS, or whole genome sequencing analysis. So a typical $2,000 GPU card now offers up to 2,500 parallel processing cores, as well as a large amount of onboard RAM, up to 12 gigabytes of dedicated onboard RAM. And for example, our recent calculations show that GWAS analysis in 1,000 Human Connectome subjects with full 1 million genotypes only takes about 5 seconds on one GPU card, and with 2 GPU cards you can actually cut it down to about 2.5 or so seconds, so it's really, really fast. And again, this is how to download solar eclipse. And if you have something that's outside of the solar eclipse functionality right now, don't hesitate to contact me and to discuss the project, and there's a good chance we'll try to accommodate your project with our development. And I'd like to acknowledge our collaborative team, which includes John Van Gero, Charles Peters, David Glant, Tom Nichols, Bennett Landman, and two of my Enigma collaborators at Nader Jahanshad and Paul Thompson, and support from NIH. Well, yes.
So I'm slow on the reaction time here today. So going back to your first talk, actually, in terms of transforming variables, inverse normalization or whatever it is, it looked like you could take a variable that was enormously positively skewed and make it normal. And I'm thinking actually of like count data and stuff with a lot of zeros. Can you deal with that, or do you need some different, Poisson type parameterization, or what do you do?
This is a really good question. So if you have a distribution that can be mapped to the normal distribution, the inverse normal will work by default. However, there are some distributions that cannot be
Like
mapped.
symptom data, for example. We have all this, hand symptom data, and half the subjects have a 0 for any given, dimension.
But again, you see, in my experience, the only distribution that's difficult, so a 2-hump distribution can actually be mapped to the normal distribution. So if you just have like one hump at 0 and the other hump somewhere, that can actually be mapped to the normal distribution. But tri-hump distribution gets to be difficult, especially if you have a big gaps between them.
And what about excess zeros or whatever that, so not 2 humps, but really a Poisson kind of distribution?
I think we'll have that needs to be looked on a case-per-case basis. Like for example, I was very successful in analyzing flare lesion data, which is very much like you said. You have a lot of subjects who have 0 flare lesions, and then you have subjects with some flare lesions. And when I performed inverse normal normalization, I actually observed that it did flatten it out into the Gaussian shape, and basically you had a lot of subjects in that end of the distribution. I mean, what you're describing is a very basic statistical model, the statistical problems. And there are standard ways of dealing with it. Like, I think one way of dealing with it is instead of having a 0, you basically add a very small random value to it. And so you basically make it continuous, but not very meaningful. But that will map to the normal distribution relatively well. Well, what Solar does, it actually saves the inverse normalized residuals. And you can basically load them up in Excel. It saves them in the CSV file. And you can open it with Excel and then plot the histogram. I commonly do it on the traits on which I'm not sure about. And most of the time I see that the mapping can actually be performed. I mean, it may not be particularly meaningful mapping, but it will be performed with 0 data, basically concentrating in that end of the Gaussian form, and then another set of data concentrating there. But generally speaking, it's best to ensure that the data can be mapped to the Gaussian form, either by means of this clever technical trick, or by sort of knowing that there is a transformation that will do it for you. Yes?
I'm really interested in the integration of the imaging data. And maybe I went a little fast with it, but you can do it on a voxel by voxel basis.
You can do it on a voxel by voxel basis. In fact, this is the most robust way to do it. So, I mean, even though we have a nifty file reading technology, in 90% of the time when I'm asked to help with a voxel-wise project, I basically tell the site to save the data in a large CSV file, where basically we have tools for doing that. Reason for that is very simple. So right now we are using nifty file as our file format, and unfortunately it's very memory intensive format. So when you have a data set, it's saved in this cubic matrix where 20 to 50% of the space is empty, but it still takes the space. So you have to load, by definition of genetic analysis, you have to load all your data into memory. You have to have it in the memory. So unless you have parallel, and typically you execute it on the parallel cluster, because you have to do 100,000 analysis. Typically you execute it on the parallel cluster, and to execute on the parallel cluster, each of your nodes have to have sufficient memory to read the entire dataset in. So this can be accommodated by two means. We have a program that will take a 4D dataset and subdivide it into datasets of slices. So for example, you would save 5 slices per subject, and then it creates a set of 4D files, and then it assembles it. Or we have a program that will read a 4D file, and then you specify how many output CSV files you would like to have, and typically you just say, well, the number of output CSV files is equal to the number of nodes on your cluster. And then it basically splits them into those, in the equal number of CSV files, and then you submit those CSV files separately to the cluster.
That cluster would have to be defined a priori.
Well, cluster is basically, SGE, but you have to know how many nodes you actually have on your cluster. And then you basically say, well, I want, I have 10 nodes, so I want 10 files. Or I have 20 nodes, so I want 20 files. We want to know the number.
But it's not sampling each voxel or whole grain level, looking at heritability, and then outputting some sort of differential heritability estimate across the grade, for example.
No, what it does, that would not be very meaningful because, heritability on the voxel-wise level can be very different from the heritability on the whole genome level. But what it does, it stores the parameters from last several fits, and then it uses those parameters from the last several fits as the preliminary parameters for the next fit. And so instead of doing 30 or so iteration for the maximum likelihood fit, you actually only do maybe 5 to 6 iterations. So it speeds it up dramatically because the heritability in neighboring voxels are typically very, very similar. And so the parameters are typically very, very similar. And the disadvantage, of course, it can crash and then it has to restart again, but it takes longer. But there are two developments that will drastically simplify everything. So in collaboration with Human Connectome project, we are working on a new genetic imaging file format. And that genetic imaging file format will take the NIFTY file and save it into a file that is going to be specifically optimized for genetic imaging analysis. And this will be, we are trying to do it proper, so we are establishing the committee and then we'll submit it to the format registration committee and all this. And the same protocol, the same format will be, this format is important because of the fourth part of this workshop where we are talking about great speed up. Because what we will do is, we will take the trait data and then we will transform it in a way that will allow for very fast genetic analysis and estimation. And to do that, we basically need to mark transform versus non-transform data set, as well as we need to store the vectors used for the transform data set. So when you combine multiple data sets, the solver will ensure that they are in the same EVD space. Otherwise, this can create a really, strange, it will basically can create havoc. So I think in a year or two, we may actually discontinue NIFTY functionality altogether and call it, I think we're going to call it CIFTY, which is like Connectomics Imaging Genetic Format. And there will be just some nice tools for converting NIFTY formats and GIFTY formats to that file format. And the same format we will plan to reuse for genotypes. So now instead of saving the data in CSV or CSV.gz file, we will convert those data sets into the same format, and then it's going to be really optimized for high performance computing, because high performance computing just requires high performance file format. Otherwise, you spend all your time reading and writing data to a network hard drive, and that's not very high performance. All right, well, I'm going to give this